Hello, this is Brian Kinghorn with the second in our series of videos on genetic markers, and today we're looking at managing individual marker loci. So in the previous video we looked at coding markers in the main data file so you can have your information come into Matesel. In the next video following this one we'll look at managing sex-linked markers, and that will be interesting to a few of you, but most of you will probably skip that one. Then we'll look at handling multiple recessive lethals. That's potentially hundreds of loci, and that provides a route to managing multiple gene edits, so that might be a video interesting to those of you working with gene edits. And lastly, we'll look at setting up and using the genomic relationship matrix for managing genomic diversity and genomic inbreeding. Just to set the scene a bit, what do we use individual markers for? Well, that's to exploit the direct effects of these marker loci, and that's through existing polymorphisms such as major genes, QTL, but also new polymorphisms that we've created, gene edits, and depending on how these have been generated, they may be segregating in the population, and Matesel can be useful to you to help break these through probably over a number of generations. We also have the case of exploiting the linked effects of individual markers that by themselves have no effect but are linked to something that does have an effect. In that case, it's better to use genotype probabilities for the underlying QTL if you can do that, or genomic selection, but of course you can still go ahead and use linked individual markers. And the next two categories are not really about individual markers but the collective effects of markers, and firstly using the genomic relationship matrix derived from multiple genetic markers. We'll use that in a later video to discuss managing diversity and inbreeding. And finally, of course, we have genomic EBVs. Of course, Matesel handles these, but Matesel doesn't create them, they're read into Matesel. So in this video there are three parts basically. We're going to first of all look at the layout and navigation of marker histograms, and there could be rather many of those histograms. Then we're going to look at managing allele and genotype frequencies for each of these marker loci. And finally we'll look at saving and editing default settings so that you can run these routinely or possibly use them in simulation work that runs over many years into the future. Let's look at an example. We'll use Matesel Classic Demo, and in the last video you saw how these codes are used to represent either genotype information as integers or genotype probability information for the different marker loci we've got. When we make that run, this is the sort of thing we see. We've got histograms for progeny inbreeding, individual coancestry, the traits, and indeed the markers. Let me talk about the layout on this page. We have got 4 columns for the 4 marker loci in the data set. The first 2 are genotypes and the second 2 are from genotype probabilities. Then the top row shows you the allele frequency of one of the alleles, the first listed allele, and I usually use the convention that that's a favorable allele, but of course you can do it any way you want. The next 4 rows show the genotypes. For a biallelosis, of course, there are 4 of these: 1, 1, 1, 2, 2, 1, 2, 2, or however you name your alleles. These are all distributions of what we expect to find in the progeny given the matings that we've got in the current solution. So let's look at an individual that is certain to be of genotype 1-1 in the progeny because its parents are both of genotype 1-1. So that individual will have a value on the allele frequency scale of 1. So what we're looking at here is the allele frequency within the individual, and that is 100% 1. If it were a 2-2 individual, it would be 0% 1, it would be in this column. So our individual with the genotype 1-1 will be represented at the end here for 100% frequency in allele 1. It would also be here because it's 100% certain to be of genotype 1-1, and it would be represented in the first column here, 0 chance of being 1-2, 2-1, or 2-2. So that shows you how these histograms get filled. Because we're looking one generation ahead in the progeny, we don't have certainty of the 3 different genotypes, and that's why we have 5 columns for allele frequency and in fact 4 only for the genotype frequencies, and the reason for that is described in the last video. So to complete this section on layout, what happens when we have more than 4 loci? Well, the maximum shown on the marker window is 6 loci, as you can see here. If you have more than that, then you can pan across to the right. So what you need to do here is somewhere to the right of center, you hold down the Alt key and click left, and that will bring you to the next set of markers. Now we're looking at 7 to 12, and we can keep going. In this case, we've got 100 marker loci. Now these are putative recessive lethals, and that's why their frequency is very high for the favorable allele, but you can see locus 43 has quite a number of individuals that are not fully certain to be of the favorable allele genotype. But you will see that there are very few of the 2,2 recessive lethals expressing mortality in utero, and that's probably because we're selecting against them, as well as, of course, the fact that the allele frequency for the 2 allele is very low indeed. And when we click on the left by holding down the Alt key and clicking, we move backwards to the first listed loci. And here we are back at marker 1. Now we'll move on to look at how you manage these distributions to give you the sorts of outcome that you want. And in the manual, there's a section on how to manage outcome distributions, not just for markers, but for all the other components. And we'll deal with that rather quickly here in this video. So if we click left on a distribution, this window comes up and we can choose amongst a number of different control types, as we call them. Let's say we want tactical desired gains. And that means that we want the mean of the distribution, which is this red tick, to come, let's say, up to this point here. And I'll click here to invoke it. I'll give it a weighting of 10 so we have definite things happening, so that this has a rather dominant effect in the analysis. And rather quickly we see that the mean of the distribution has come to where we want it to be, probably around 0.625. This is a bit difficult to see, so what you can do is hold down the Shift key and click left, and you'll get that picture to fill the whole screen. But I'm going to do the opposite and hold the Control key and click left and go back here so that we can see what we're doing with more than one distribution at a time. Now let's say this is a recessive lethal or an unwanted recessive effect, and 0.625 is part of our plan to fixing this allele in the long run. But in the meantime, we might not want to have any expression of this effect, so what we will do is decrease the mean for the 2,2 genotype. I'll also give that quite a strong impact, and within a short period of time, Matesel's given us what we've asked for these two components to get to this frequency of the favorable allele and have no expression of the 2,2 genotype. But of course, this may be at too much of a compromise, and we can start reducing these weightings until we get what we want overall, given what is possible. In the old days, before we had DNA tests for these things, we would not fully do this. We wanted to have certain expression of the unwanted effect, because that was where you got the information for what the probable genotypes are for the individuals involved. Here's another example in the manual that you can read about, and in this case we've used the bimodality tool in order to target 80% black calves and 20% red calves in this breeding operation. And lastly, and quite quickly, what we can do is click the save icon up here to save the settings that we've made in these marker manipulations. And when we bring that across with Matesel ini, we can see that for marker 1 we have invoked control type 12, which is the desired gains, with a weighting of 10. And this was where we clicked at 0.6413 is the actual frequency. And we also asked for the genotype 2,2 at marker 1 to be invoked with control type 2, which is to decrease the mean, also with a strong weighting, probably too strong. So this is useful for when we want to actually apply policy and have it set up immediately in Matesel when we start to run. And as I said before, you can use that for routine use, or it's also useful in simulations, and we'll describe that elsewhere. So that's us at the end of this video. The next video will be on managing sex-linked markers, and thank you for listening.